My background is in environmental chemistry and environmental engineering and public health. At Silent Spring Institute, our mission is to understand links between everyday chemical exposures and our health. We got started studying water quality on Cape Cod due to concerns about high breast cancer incidence and questions about whether there were links to water quality or other environmental factors. For about 20 years, we've been thinking about emerging contaminants and water quality. And Cape Cod is a region mainly served by septic systems, so it's very pertinent to the topic of today's conference. So today I'm going to introduce you to the topic of contaminants of emerging concern, where they come from, why they are of concern, talk briefly about some of the water quality challenges facing Cape Cod, talk a bit about our water quality research, which I could talk about for hours, but I will try to stay within the time limit. And then Chris specifically asked me to think about how our work could tie into questions about what are indicators of septic system impact. So contaminants of emerging concern, or emerging contaminants, is really a broad term, and there's no set list of what exactly is on this list or what makes it exactly emerging. But here are some examples. If we think about all the products that we use in our home, in our domestic wastewater, there's medications and cleaners and all kinds of products that we have. I'm an environmental chemist, so I look at this and I think of all the chemicals that all those products contain that end up in our wastewater. So hormones and pharmaceuticals, antimicrobials, detergent ingredients, highly fluorinated chemicals and things like Teflon and Scotchgard, flame retardants, artificial sweeteners. These all end up in our wastewater and end up in the environment. So I'm going to parse apart the phrase contaminants of emerging concern. So these things are considered contaminants when they show up in drinking water or groundwater or surface water. So it's not a contaminant when a pharmaceutical is in your medication, but we consider it a contaminant if it's in your drinking water. For the most part, these chemicals are considered emerging due to improved analytical capabilities. In recent years, scientists have new methods to test for parts per trillion and even lower levels of chemicals. So the analogy I like to use is of looking at the night sky with your naked eye and you see a certain amount of stars. And now if you go to an observatory and you have a high-powered telescope, you see a lot more. Of course, all those stars were there all along. You just can see more with new technology. In some instances, there are new emerging chemicals because we know that new chemicals go into use every year. These are loosely associated as emerging because they are not currently regulated in drinking water. There are questions about what makes something emerging. There are things that we call emerging contaminants that have been of concern for decades. So it is kind of a loosely used term. Someone was saying it was contaminants of emerging regulatory concern. And then why are they of concern? I'd say from the ecological perspective, the links are a lot clearer. It's easier to study how chemicals can affect fish and other aquatic organisms. A lot of the concern about ecological health effects focus on endocrine disruptors or hormone disruptors. For instance, chemicals that mimic or are hormones can cause feminization of male fish. So for instance, in the Potomac River, 80% of male smallmouth bass were considered intersex, meaning they had male and female characteristics. In terms of human health, many of these chemicals have been linked with human health effects at high levels of exposure, but we really don't have a lot of information about what are the health effects of kind of chronic, low-dose exposures to many different chemicals all at the same time. So how do emerging contaminants get into the environment? We think a lot about household wastewater as a source of emerging contaminants. So in many places that means there's a wastewater treatment plant that's a source. On Cape Cod. So we have wastewater treatment plants, septic systems, wastewater from hospitals and other medical facilities. There can also be things like veterinary antibiotics, so agricultural runoff or runoff from livestock operations. So there's many different pathways. I'm going to focus on septic systems today, given the topic of the summit. And again, on Cape Cod, 85% of residents have a septic system. So this is very pertinent to water quality concerns. So we got started with our research on Cape Cod addressing drinking water quality concerns. And right now on Cape Cod, the big challenge, we've heard about nutrient pollution here in Lake George. Right now on Cape Cod, nutrients are a huge concern, and particularly how to control nitrogen discharges into coastal waters. It's kind of reached this tipping point where people can't ignore the problem anymore. And there's a lot of emphasis on how can we address nutrient pollution, not just with sewer systems, which aren't really practical in some places or are too expensive, but what are alternative tools that communities can use to protect their watersheds. So it's an important problem. It's getting a lot of attention. I do feel like the communities have put kind of their blinders on and just are thinking about nutrients, and we're out there trying to say, well, there are all of these other chemicals that are in that same water if you're seeing high levels of nutrients. And if you're thinking about making these big investments in your infrastructure, you don't want to have to do that again in a few years if we start regulating some of these emerging contaminants, or we realize we're not adequately protecting the drinking water as well as coastal water quality. So I was happy to see that the Boston Globe magazine did have a big story last summer about problems of emerging contaminants and other threats to drinking water quality as well. And again, some of the challenges specifically for Cape Cod are that 85% of homes have a septic system. We have a shallow sand and gravel aquifer, so there's a lot of potential for contaminants at the surface to get into the groundwater. Since the 1950s, there's been rapid development on Cape Cod. And all residents rely on groundwater as a source of drinking water, mostly public water supplies, but also about 20% of residents on private wells. So our research on water quality on Cape Cod has focused on measuring levels of hormone disruptors and other emerging contaminants in drinking water, in groundwater, understanding how those contaminants move through the aquifer, and in ponds. So in Cape Cod, we have many kettle ponds, and these are mostly fed by groundwater. And so they're considered windows onto the aquifer because they kind of can tell you about water quality in the aquifer and integrate the signal of the upgradient land uses. We've evaluated septic systems as sources of emerging contaminants and characterized how they move through the groundwater. And again, we're trying to inform the decision-making on the Cape, all these decisions about wastewater management and drinking water protection, to consider these other contaminants. Even though they're not necessarily regulated now, we need to have these on our radar screen. So septic systems are not designed to remove emerging contaminants. They're mainly designed to remove pathogens and solids. If you're lucky, you can get some enhanced nutrient removal. These were not intended to remove emerging contaminants. Some will break down or be removed anyways. I'd say you don't get too much removal in the septic tank, although certain chemicals that stick to particles could end up in the sludge. But in a drain field or leach field, you can see removal of some of these emerging contaminants through a combination of biodegradation, so the microbial community can break down certain contaminants, through sorption, so chemicals that stick to the soil particles. And then for a handful, you might see volatilization, so fragrance compounds could potentially end up in the air space, the pore spaces. But it's really sorption and biodegradation that people think about. So if you have a functioning leach field, you will have pretty good removal of certain emerging contaminants. If your leach field's not working well, or you have an older system, then you'll have poorer removal of emerging contaminants. And so the ones that don't break down will be transported to groundwater and can end up in surface water or drinking water. So I'm going to touch on just some highlights of our past water quality research. We did a study of ponds on Cape Cod in 2008. We compared 3 ponds in more densely developed areas, ponds that we thought would be more impacted by septic systems, and 3 ponds in less densely developed areas. And we wanted to see whether the levels of hormones and pharmaceuticals differed between those 2 types of ponds. So what we found was that, not surprisingly, if you have more residential development near your pond, more septic systems, you're likely to have more frequent detections and higher levels of hormones and pharmaceuticals. So what I'm showing here are the levels for certain hormones and prescription medications that showed up. These are the ponds located in more densely developed areas, and these were better protected. So you all know that keeping land open around your pond or your lake is going to lead to better water quality. Another way of showing this is to show the highest concentration that we detected for these various chemicals. Again, you see the highest levels tended to be found in ponds in more densely developed areas. From an ecological point of view, the fact that we were finding hormones, this is at nanograms per liter or parts per trillion levels, so these are what we would consider fairly low levels, but for ecological health, having these parts per trillion levels of hormones can have implications for aquatic species. The highest level we saw here was about 4 to 6 nanograms per liter, and some studies in fish in the lab have shown effects at 10 nanograms per liter. So we're kind of entering that range where there could be ecological health effects. And then we turned our attention to drinking water quality. So we conducted a pair of studies focusing both on public and private drinking water wells. And our goal in selecting the wells was to include some that we thought would be among the most impacted, some with some moderate impact, and some that were in more pristine areas to see whether we also saw that reflected in the levels of emerging contaminants in these wells. We tested for around 100 different types of emerging contaminants, so pharmaceuticals, personal care product ingredients, hormones, flame retardants, fluorinated chemicals, many different types of chemicals. The type of testing that we do is pretty expensive. It cost us about $2,000 a sample to test for all those different types of chemicals. So this isn't something that's going to be part of routine water quality monitoring. So we also wanted to get a sense of whether we could identify indicators of emerging contaminant impact. So we evaluated land use, we looked at nitrate and other chemical markers of septic system impact. And then we also wanted to compare our results on Cape Cod to what we knew about levels in other drinking water supplies in the US to see if this confluence of factors on the Cape did lead to relatively high levels for certain contaminants. So again, I could talk about this for a long time, but I've tried to make this brief. So the types of chemicals that we found most frequently included an artificial sweetener called acesulfame. This is used in certain diet sodas and low-calorie yogurts and juices. We found certain antibiotics and other prescription medications. We found flame retardants, for instance, chemicals that are used in foams in people's houses. These can end up in people's dust and eventually make their way into water, either through laundering or maybe through mopping your floor, and highly fluorinated chemicals that are used in nonstick, stain-resistant, and waterproof products. The levels that we found were typically in the low parts per trillion or nanograms per liter range. Many of the chemicals we tested for we didn't find, or sometimes we found them at lower levels. But when we did find these chemicals, those were the types of concentrations that we found. And we found that some wells in less densely developed areas had none of these or just 1 or 2, and then the most contaminated wells had up to 15 different chemicals. And then we did see for a couple of the pharmaceuticals that the highest levels that we found were pretty much as high or higher than had been found in other studies of drinking water quality in the US. So this confluence of factors, having these septic systems, having this shallow aquifer, does lead to relatively high levels in some cases. So, okay, so we found these things at parts per trillion levels, like should we worry, what are the health implications? And it is important to keep in mind the order of magnitude that we're talking about here. So in terms of a therapeutic dose, if you're actually taking a medication, the amount that you would ingest is millions of times more than you would ingest if you drank water that had these chemicals at parts per trillion levels. And it's also important to keep in mind, and sometimes the drinking water suppliers say, why are you always picking on drinking water? People put use nonstick pans, they put stain-resistant carpets in their house. You know, water is not necessarily going to be their main route of exposure. Which I think is a fair point, but I think there's also something to be said for protecting the water resources, because once chemicals get into the environment, you can't bring them back in if you decide to change the chemicals that are in use. And we're also thinking about parts per trillion or nanograms per liter level. So we've really amped up the magnification, and we're looking at lower levels. And we're used to thinking for other types of contaminants about parts per billion or micrograms per liter levels. So we're finding a lot of things, and it's important to note that just because we found it, it doesn't mean it's of concern. But we do think that the presence of these types of contaminants is worrisome and does merit further study. In the case of medications, we know that we don't give certain medications to children or to pregnant women because the developing body is more sensitive. So we're not supposed to be exposed to these types of chemicals all the time. We know that there's side effects that can occur at levels below the therapeutic dose. And we also know that some people are more sensitive than others. Think about people who are sensitive to certain antibiotics. We're still learning about potential synergistic effects. So the idea that being exposed to multiple chemicals at the same time can produce effects that you don't see when you study each one separately. And in the case of endocrine disruptors, there's new research showing that in some cases, you can see health effects at low levels of exposure that you don't see when you look at higher levels of exposure. So usually with toxicology, we dose animals with a high dose, and we make some assumption about how to extrapolate to lower levels. And for endocrine disruptors, we might be missing some effects. So like I said, we also thought about what are indicators of these emerging contaminants in the public and private drinking water wells we tested. So that this is an example from our public well study. We binned the wells into 3 categories, ones that had less than 0.5 milligrams per liter, 0.5 to 2.5 milligrams per liter, parts per million, or over 2.5. And this was the average number of emerging contaminants detected in each of the wells. So you see that where there's more nitrate, you have more emerging contaminants. My background's in engineering, so I had to put in some numbers here. So this is, these are correlation coefficients. This was from our private well study, where we did statistical associations between certain factors, so nitrate, boron. This is residential density in the nearby area, which we did with GIS, the artificial sweetener, acesulfame, and well depth. So we evaluated all these indicators, and then we looked at how well they correlated with both the number of detected emerging contaminants and the total concentration of pharmaceuticals. So we weren't necessarily interested in, is it sulfamethoxazole or carbamazepine, or which exact chemical is there, but what's sort of the overall signal from pharmaceuticals. So walking through these, so both nitrate and boron are inorganic markers of wastewater. And in terms of what these numbers mean, that's just showing the strength of the correlation. So the closer the number is to 1, the stronger the correlation, and I've shaded in bold the ones that are statistically significant correlations. So we see that both nitrate and boron are associated with the number of detected emerging contaminants. It seems boron is a better, more specific marker. And having both together can be helpful, because in some cases you might see high nitrate from fertilizers. For a few of our wells, I think we saw high boron because of potential saltwater intrusion. So having both together helped us kind of tease apart which ones had the greatest wastewater signal. We looked at nearby residential density and saw some association, although not as strong as the chemical markers of septic system impact. And in terms of the 27 different emerging contaminants that we detected, the artificial sweetener acesulfame was kind of the best marker for the presence of the other contaminants. It tended to be present at the highest levels, and it was also the case that if we saw any other pharmaceutical or flame retardant or fluorinated chemical, that artificial sweetener was always there. So that was kind of the one chemical that was probably the best marker for the other ones. And then we also looked at well depth. So we expected that shallower wells would be more vulnerable to emerging contaminants. So we did, in fact, see that effect. The negative sign here shows that deeper wells tended to have fewer emerging contaminants and at lower levels, which we expect. So again, Chris asked me to sort of put this into a context of thinking about how these chemicals might be used as indicators of septic system impact. So starting with the inorganic compounds like nitrate and boron, I'm going to kind of go through the pros and cons that I can think of for all these different types of contaminants. The good thing about nitrate, or maybe you'd want to look at total inorganic nitrogen, is that it's inexpensive, it's pretty easy to measure, and that, at least in aerobic conditions, it doesn't degrade. The problem is that it's not necessarily very specific to wastewater, because it can also come from fertilizers or atmospheric deposition or other sources. And there's the potential for biotransformation and biological uptake, so it may not behave in a conservative or non-degradation way. Boron is actually a pretty good marker, so boron's used in a lot of laundry detergents and soaps. It's inexpensive. It's fairly easy to measure. And if you're going to go to a commercial, so in terms of going to a commercial lab, these would probably run you about $20 to $30 per sample, not hundreds of dollars like we saw with emerging contaminants. And many labs would have the ability to measure boron. It doesn't degrade, and it's fairly specific marker for wastewater. I put here multiple sources because we can't rule out that there might be other sources, and there might be sort of variations in what's considered a background level. So then there are contaminants of emerging concern, things like caffeine or acetaminophen, which is in Tylenol, which show up a lot in surface water, but may not be the best indicators of septic system impact. They are specific to wastewater, and they're present at fairly high concentrations. But they are fairly well removed in leach fields, and they're not all that persistent. So it's kind of a pro and a con. I guess if you're looking for, where we're seeing septic system overall, you might miss it because they do tend to break down. On the other hand, it might help you distinguish between areas where septic systems are failing and where they're sort of working adequately. Because if you have a failing septic system or cesspools or sort of older systems, you actually might get these making their way through. So that can help you distinguish between those two. The other downside is that they're expensive to analyze. So you need specialized equipment. You need to make sure you're not getting caffeine from your morning coffee. That was one of the sad things about doing the sampling, was that I couldn't drink coffee while we were out in the field. Because we're looking at such low levels, you want to make sure the chemicals are coming from the water itself and not being introduced from the environment. So then there are other medications, prescription medications, that are fairly persistent. So I'm giving two examples, sulfamethoxazole and carbamazepine. These show up in many studies of emerging contaminants. They do tend to persist in the environment. And if you're running a method where you look at a lot of different pharmaceuticals, then you might, even if no one in your area is taking one or both of these specific chemicals, if you're testing for 100 different pharmaceuticals, you're likely to pick up at least some of the chemicals or pharmaceuticals that people might be taking. The challenge is that if you're looking at other types of prescription medications, that there is variable removal depending on what type of septic system you have. Again, some are well removed and some are fairly persistent. Usage can vary widely among households. So if you're looking in an area with a limited number of homes, it's possible that there just aren't any people in that area taking that specific medication. And again, these are also expensive to measure. Artificial sweeteners, I'd say, of the emerging contaminants we tested for, show pretty good promise as markers. So they're present in high concentrations in wastewater because they're in so many products. And there are some that don't degrade or sorb to soils. So acesulfame and sucralose in particular seem to be most persistent. Cyclamate and saccharin are less persistent. So some studies have suggested that having the two families is kind of useful because, again, if you have some showing up that break down fairly easily in a leach field, then it's suggesting that you're not getting that full treatment and there might be failing systems. And then these are more conservative. The downsides are that, again, these are expensive to measure. They're not as routinely measured by laboratories as some of the pharmaceuticals and other emerging contaminants. And our understanding of their environmental persistence is evolving. So I submitted a paper for review recently on levels of emerging contaminants in septic systems and said, acesulfame is really persistent in the environment, and they came back and said, well, here are 3 papers that show that it does break down. So our understanding of their behavior in the environment is evolving. And then the last category, we didn't test for these specifically in our drinking water studies, but we did look at them in other studies of a plume from a septic system. So these are optical brighteners or fluorescent whitening agents. So these are used in most laundry detergents, and they make your clothes look brighter, not because they make your clothes clean, but because they actually have a fluorescent property that makes your clothes glow a little bit. So if you are somewhere where there's a black light, your jeans will light up, but like your shoes won't. It's because of the optical brighteners that bind to cotton and other fabrics. So they're very common in wastewater. And the good thing is that the fluorescence is easy to measure. So you don't necessarily need a really fancy instrument with a mass spectrometer to measure these. You can sort of tune to certain wavelengths of fluorescence and get an idea of the presence of these, not for any one specific compound, but just for the presence of them as a family. And there's some cool potential, I think, for citizen science. There was actually a study in England where they used cotton tampons as basically as passive samplers. So the idea is that these chemicals stick really well to cotton. And in that case, they had a river, and they were trying to figure out where the illegal discharges of sewage were. So they deployed these in the river, and then they brought them back to the lab and put them under a black light and to see where they were glowing. So I think there's kind of cool citizen science opportunities. And again, these are unique to wastewater. The downsides are that they may have limited mobility in groundwater. We did see some degradation of these compounds in one of our studies of septic systems. They can undergo photodegradation, so if you're measuring them in the water body itself, they might degrade. There can be other chemicals naturally occurring in the water that fluoresce at those same wavelengths, so you'd have to take into account the background. And if you wanted to go beyond that and measure specific fluorescent whitening agent number 1 or number 2, then again, those would be expensive to measure. So I guess in summary, what I would say is that contaminants of emerging concern are a growing concern. They're not regulated currently, but they're really on the radar screen, and there's potential that down the road, at least for drinking water, that they may be regulated, and there's a lot of concern about potential ecological health effects. I'd say in terms of treatment that there are options to enhance removal for some emerging contaminants, some enhanced septic systems, or if you have a wastewater treatment plant, you can tweak things and you can improve the removal for some, but others will be persistent pretty much no matter what you do. And the message that we've tried to put out on Cape Cod, and I think it applies here too, is that if you're seeing high levels of nutrients or coliform bacteria, you probably have these other chemicals there as well. In terms of thinking about indicators of septic system impact, the emerging contaminants themselves are pretty expensive to measure, so probably they wouldn't be part of a routine monitoring program, but they might be useful in certain individual cases where you're trying to figure out what the source is. It might help you tease apart where some of those other pollutants are coming from, and could be used in tandem with boron or with other more easily measured compounds. So with that, I just want to thank all the people that contributed to the work that I presented today and funded the work, and a little bit of see we really do the work. Here's some members of our team and volunteers. And if you would like to learn more, we post all of our papers. We have fact sheets and press releases on our website at silentspring.org/water. And my email address is off the bottom of the page, but I'll be here all day, so you can feel free to find me if you want to talk more. Well, thank you.